I believe I've been given the signal that we can start, and thanks everybody for joining us. This is our first virtual town hall, if you will, since the COVID epidemic began. And the intent is to inform you about things that we're doing within the Flint Animal Cancer Center and how COVID has changed or not changed our demeanor and our functions. First, I'm not Chris Wallace. And we are, in fact, socially distanced here, so we don't need masks. We're at least 8 feet apart, best we can tell. I'm Steve Withrow. I'm one of the co-founders of the Flint Animal Cancer Center, and spent a career as a surgical oncologist, mostly at Colorado State University. And as most of you know, personally or with your own health or your pet's health, cancer has not gone away. And we continue to fight COVID and cancer at the same time. I consider it a good omen that today happens to be Lucy New's birthday. Lucy was a very informative, courageous patient of ours. Her owner, Jeff, out in California, I think has been deployed with the California fires, but this is good for us that this is Lucy's birthday. The format will be pretty simple. I'll introduce quickly the three speakers, and then ask them to talk about their specialty functions within the Flint Animal Cancer Center before COVID, and then what's changed after COVID. So first on my left is Dr. Rod Page. Rod's I should preface that by saying I looked at their resumes, their CVs, which I should know by heart. But collectively, I've had 57 years of collaboration with the three people on my left. Amazing, if you count vet school through residency through faculty positions. So first, Rod Page is a CSU grad, College of Vet Med and Biomedical Sciences. Did his residency at Animal Medical Center in New York, went on to build cancer programs at North Carolina and Cornell. And I was fortunate enough to get him to take my job here at CSU about 10 years ago as director of the Flint Animal Cancer Center. Sue Lana, also a CSU grad, went on to Texas for an internship, back to CSU for residency, and is currently the Stewart Chair in oncology at CSU. Rod Page, by the way, is the Withrow Chair at CSU. Doug Thamm is a graduate of the University of Pennsylvania, went on to residency at Wisconsin, and has been a faculty member here since that time. He's the Barbara Cox Anthony Chair in oncology. All three are medical oncologists. I'm the lone surgeon at the table here, and all of them are full professors within the CSU. So with that, I'm going to ask Rod Page to give us a little bit of an oversight and overview of how the Animal Cancer Center has functioned before and after COVID.
Thanks, Steve. And thank you all for joining us today. It's a real pleasure to be actually talking in real persons instead of joining you on the screen. But prior to February and March, the Cancer Center was in full mode, rolling out mode. We had a very robust clinical program, research program, a teaching effort that spanned all 4 years in graduate studies as well as residency training programs. And then we had to pivot very quickly. And at that time, the management team, the leadership management team of the Cancer Center began to meet on a weekly basis to predominantly think about what the impact is going to be for our students and for our clients and patients related to having to reduce our activity levels and our teaching format. So I'd like to basically just give you a little bit of context of how much we were thinking and what we were having to do in order to try to come up with a contingency plan. At the time, when we were told that we would be shutting down, it was projected that the Colorado State University as an institution would have a significant shortfall in funding from state funds and from other funds. There was a lot of worry that there would be reductions in tuition revenue, reductions in research, reductions in client revenue through the hospital, and we really didn't know exactly what to expect. And it took some time to figure that out, but in the meantime, we were very, very active trying to review all of our funds, all of our activities to make sure that we focused on the things that we thought were most important at the time. And that was preserving our personnel and making sure that our patient assistance capabilities were preserved throughout this period. And we didn't have any idea what the length of time was going to be or whether or not there would be a return to any sort of function at the time. So as it turned out, the university, through the Board of Governors, was able to reduce the overall deficit from an 18% deficit shortfall in funding for this for the rest of fiscal year 20, which ended in June 30th of 2020, to a mere 6% for the university, and that was actually 3% at the college. That was accomplished through both national and state support, as well as rainy day funds that were put aside by Dr. Frank over the course of the last 10 years in response to the previous recession. So we were very, very fortunate to be able to think that we would only have a relatively small type of shortfall to overcome. But nonetheless, we were able to come up with a contingency plan where we felt like we would be able to, regardless of what happens, preserve our personnel for the time being through the fiscal year, and going forward we shall see. But we also focused our work on making sure that there was plenty of patient assistance funding for clients that would come to the hospital. We've always been involved with those types of fundraising efforts, but this was a particularly critical time in order to reaffirm our priority for that particular area. So I'm going to pass it over Dr. Lana. Maybe you can talk a little bit.
Well, Rod, if I could interject,
Sure.
Can you talk a little bit about some of the national initiatives that you've been involved in? That you represent the Flint ACC, but you're also the leader in a number of national initiatives.
Sure. On a national level, I think it's everybody's opinion here for sure, and across the country, that CSU has been a leader in the field of veterinary oncology, and I felt like it was an obligation for us as an institution to reach out for a little bit broader context of leadership in the field of comparative cancer research. So over the last few years, I've been involved with a couple of initiatives. One is a research consortium that actually includes now 12 other academic institutions, both human medical cancer institutions and veterinary academic institutions, that has been formed and has been the recipient of financial funds from the V Foundation, a foundation that supports cancer research on a national level. They decided that they would create a program within their organization to help support comparative oncology. And we are in the midst of the first evaluation of projects from that organization. They have made available $2 million on a competitive basis. So it's really a remarkable group, and a really impressive sort of an effort to go from a traditional sort of a concept to a true focused comparative oncology program. I've also been involved with the National Academy of Sciences to establish and produce a workshop. We did one in 2015, and I've always thought it'd be great to go back and do something else. So we have received approval for a workshop on how environmental carcinogenic exposures would impact companion animals, and dogs in particular, to influence their development of more rapid aging and cancer susceptibility, and particularly in application to humans. So we've been able to raise a substantial amount of money for this workshop, which is required, but having the respect and the ability to say that the National Academy of Sciences has validated this concept and will produce and allow us to use their name has been a real inspiring concept as well for me.
Thank you. Dr. Lana is in charge of our clinical program, and we'll talk about our clinical operations and teaching and how it's changed with COVID.
Thank you, Dr. Withrow. I'm Sue Lana. I'm a medical oncologist, as was said during the introductions. And as I think back to March, when this all started for us or changed our world on the clinic floor, it seems like a long, long time ago, but actually, 6 months ago. So from a Friday when operations were normal, we see roughly 6,000 patient visits per year, to Monday when our clinic was then shut down, and the whole hospital at the veterinary teaching hospital was shut down and was then seeing emergency only cases. So we quickly, those of us who showed up to work that day came in, called clients, canceled appointments, rescheduled appointments. We didn't know how long the shutdown was going to be. Quickly then found out within a couple of days that the percentage of people who could come into the building was also slashed by about 50%. We had to come up with plans according to state regulations, and then the university shut down as well, about how many people we could have in the building at any given time, seeing the patients that we were currently seeing. Then the governor's office came out with an order that elective procedures could not be done and that only emergent procedures and surgeries and things could be done. So for us, cancer didn't stop, and we were trying to figure out what should we do about our ongoing chemotherapy patients, patients who really needed to be seen, ongoing radiation patients. And so we took some guidelines from the National Comprehensive Cancer Center Consortia on the human side, who had some very nice guidelines come out really quickly in March about chemotherapy patients and their need to continue treatment. And so we provided these guidelines and our plan to our hospital administration. They allowed us to go ahead and move forward with seeing these cases because we felt it was really important that our patients have continued care. We decreased our personnel pretty quickly, including faculty and residents. We altered our scheduling and prioritized the patients that had to come in on an immediate basis and started kind of working through the flow. For patients who we had to postpone, we stayed in touch with them as best we could and got them in as we could over the course of the next couple of months. Currently, as caseload has increased and restrictions have been loosened from stay-at-home to safer-at-home within our state, we've been able to have more and more cases come in. Around May period of time, we were up to about 75% of our caseload, and we're now back up to more of a consistent caseload in the hospital. So we feel pretty good about that. From a teaching perspective, same sort of thing. The students were with us one day, and we see fourth year and third year veterinary students on the clinic floor with us, and they help with all of our patient care and interacting with clients and things. They were told on a Monday not to show up anymore until further notice. So we, in addition to figuring out how to function without our students, we also then had to pivot, which is our favorite word these days with the pandemic, into virtual clinical teaching. And so coming up with ways that students could join us in rounds. We all learned very quickly how to use Teams. We all learned what worked, what didn't. We came up with clinical teaching modules that the students could do at home on their own. They were sent surgery kits and surgery mock-up things for them to do at home to learn their suturing skills. And continued down that pathway, graduated a class of students in May, and then had students come back into the building with us at the end of June. So that's kind of the phase we're in right now. The fourth year students are with us in the building. There's still no clients allowed in the building at this point. The junior students are coming back in sporadically, and everyone wears protective equipment all day long. We all have masks. We all have eye shields or face shields if we're working within 3 feet of each other. We all have outer garments that we wear. And so the personal protective equipment that's required to be in the hospital is pretty extensive, and so far so good. We've continued to be able to provide care for our patients as they come in.
Okay, Dr. Thamm is a clinician, but also in charge of many of our research initiatives that go from the laboratory to the clinic, and also oversees much of the regulation of our clinical trials program. So we'll ask Doug to concentrate on research and clinical trials.
Thanks, Steve. So in all fairness, I should say that I really share the directorial obligations for research with my colleague Dan Gustafson. So he tends to sort of oversee the laboratory research aspects of things, and I'm sort of in charge of overseeing the clinical aspects of things, but I certainly can speak about the laboratory research component as well. So as Sue said, as far as clinical research goes, our timeline was actually very, very similar to that of the whole clinic. So when things shut down in March, unfortunately, at least in the very, very short term, that exact same thing happened with the clinical trials service, but in very short order, we realized, just like with our regular quote-unquote oncology patients, that it was incredibly important to make sure that we were able to maintain care for those patients who were on a clinical trial who needed to be seen to continue their cancer care. So very, very quickly, we were able to make that accommodation. So those animals who were on a study who needed to be seen to make sure that we were able to continue to provide them the care that they needed were seen right away. As the months have gone on, we have been able to open up more and more of our clinical trials, and part of that has been guided by, again, the guidelines that have been established by the veterinary teaching hospital for how many people can be in the building, what percentage of a normal caseload is appropriate, and we were able to sort of prioritize reopening certain trials versus others based on those guidelines, based on, again, need, based on priority, based on a lot of other things as well. And again, now we're back to the point where all of the trials that the sponsors have elected to reopen are now reopened. So there are a few where, again, we have an external sponsor who's decided, oh, I think we're going to wait a little while before we start up again, for their own reasons. But we're really back to recruiting animals on all of the clinical trials that were open before the pandemic struck. Again, with the exception of a couple where, again, it's not because of the pandemic, it's actually sort of because of the sponsor. So we're raring to go. We're back to looking for patients who might be eligible for all of our studies at this point. On the research side, on the laboratory research side, very much like on the clinical side, there was a period of time very, very soon after the initial spike of cases was reported where effectively everything shut down in the laboratories. There were some critical ongoing experiments that really couldn't be stopped midstream that were allowed to continue. And most of this actually came down from the college level about what we were and weren't able to be able to do. And then about a month later, we were given the approval to reopen laboratory investigations to the tune of about 30%. So it was important that we had a plan in place for making sure that the folks who were going to be working in the laboratories were safe, minimizing the number of people who were in a laboratory at the same time or in an office at the same time, and actually a printed written plan needed to be made for each lab and then submitted for approval at the dean's level. And then probably about 6 weeks ago or so, we were able to reopen to the tune of 50%. So now, as far as the personnel goes, we're operating at around 50% capacity. I'd like to think that actually most of the folks who are in the labs are so efficient that the amount of work that's getting done is actually greater than 50%. But that's sort of still the official party line. I would argue that we've still been able to be incredibly efficient and incredibly useful. And several graduate students have actually finished their programs and graduated just in the last month or two during the height of the pandemic. So I think that's really a credit to the fact that people are continuing to be able to get this really critical research done in a timely fashion despite the restrictions that have been placed on the programs.
And kind of a little bit of an editorial, but in addition to the things you just heard about, we continue to do free consultations around the world for clients and veterinarians who have cancer or oncology questions, with well over 1,000 consults annually. We're very proud of that and believe that's our responsibility to help the whole profession and society whenever we can. I'm flunking at retirement, and I can see a lot of this from afar, but it's amazing the resiliency that I've seen in the faculty and staff and the students that have taken care of all the patients throughout this crisis. Amazing creativity in teaching and clinics, and you pull into the hospital, you get a parking lot number, and the student comes out and takes your animal away. It's amazing the faith that people have in us, and the faith that they have that we can pull off what we say we can do. So it's amazing. All the support staff, anesthesia, radiology, pathology, internal medicine, et cetera, is all in place. And it's been a real epiphany of how people can pull together in what appears to be just a disaster. Which it is a disaster, but it's resiliency that has carried us forward. So they've kind of answered, the three panelists have kind of answered, what areas they specialize in and how they've managed with COVID. And I don't know if they want to make some closing comments on what they see in the next year. I think it's Einstein that said, in every disaster there's an opportunity. And I think we've learned some good things as we've gone through this, and maybe you could reflect a little bit on what you see happening in the next year or two before we have a meaningful immunization process across the country. Rod?
Yeah, so I think what we brought you kind of up to speed to the current time. So looking ahead another 6 to 12 months, I think there are opportunities that we have to seize because of the restrictions that we have and because of the issues that we have to deal with. One of them that I've seen and heard talked about not only here but otherwise is essentially telemedicine. And our consultation service, as you mentioned, is going strong. We are in the process of redoing our infrastructure to be able to manage that. And we have a number of patient and client coordinators that have taken on a responsibility of epic proportions, as well as our students and teachers and faculty, to help our clients understand how we need to deal with this and how this is going to be different than it was. Perhaps a little bit more emphasis on how we actually reach our constituents through telemedicine would be one area. I think maybe Dr. Lana could talk a little bit more about the fact that we are poised here to be able to create a robust new model for clinical service and teaching, one that is focused specifically on students in a very controlled format. They see a case that is something they can sink their teeth into and understand. And then we also are able now, because of our ability to hire additional personnel, additional faculty, we are able to think about a high volume, high quality service that allows us to meet the needs of the service clientele or the expectations of our population. So I think there's that we will be looking for. And I'd like to just basically close by saying, what we've done is to position ourselves for the maximum amount of flexibility. We don't know what fiscal year 22 is going to look like. It could and is likely not going to be as easy to recover from some of the funding shortfalls. Hospital revenue is probably not going to be back to where it was. We don't know. We can project what we might find, but through the ability to harness discretionary funds, to look at our budget, and to think about how we can maximize the flexibility in order to support our personnel, in order to support our graduate students that are in the process of getting a PhD but can't necessarily do the work in the laboratories, and looking at all the ways that we are able to help going forward. I think our management team has been extremely fluid and extremely broad about thinking about that. So in addition to us four, the management team includes Dr. Gustafson, Lauren Mingus, Christine Hardy, Tori Kapavic, and Don Duvall.
And Sue LaRue.
And Sue LaRue, a radiation oncologist. So, I have to really thank all of that effort and all of the organizational structure and support and suggestions about how we've been able to look to get to this point, and now we're looking 6 to 12 months ahead.
Dr. Lana, what do you see as good and bad going ahead?
So to speak to kind of Rod's point about what we're changing or what we're hoping to change moving forward, we had an idea prior to COVID, brought on a new faculty member, Dr. Vickery, who might have more of a clinical high throughput focus, and then Dr. Burton, who's another medical oncologist who started actually in July. And so our plans were to have everybody come on board, figure out a way to kind of come up with more of a two-service model, a teaching service and a more high throughput service for both the residents to learn what it's like to be in a practice situation, and then to concentrate differently with our students. And then COVID happened, and what, I don't want to say the good thing, but that type of stoppage and the reason that we had to pivot and do online teaching actually made us really move pretty quickly into this type of model and to try it out in a variety of different ways. And so it kind of forced our hand a little bit, but as Rod said, it's an opportunity for us to make these changes more quickly than we may have done so in the past. Going forward, our caseload now is stabilizing, and again, we certainly want to help as many people as we can, as many pets with cancer, and see cases. I think one of the nice things going forward is we do have, and we've learned a lot about how students can learn virtually, what resonates with them, what doesn't. It's very time-consuming and challenging for us to do both, teach virtually and teach in person. And so figuring out how to distribute our workflow, the university or the college as a whole is going through a curriculum renewal process and changing how vet students are taught. That also is being pushed forward because we were planning an awful lot for that, but now that everything's been disrupted, they're moving farther, more quickly ahead. And I think it's a really good time for them to concentrate on that. So I see some of the changes and some of the new things and the new ways that we teach students and residents will be things that will carry forward from this, I think. They're not bad things. We jumped into them because we had to, but I think they'll continue.
And Dr. Thamm, same question. What do you see going forward that affects research and or clinical trials?
So I think one of the things that's been kind of a happy byproduct of this is during this period of time where maybe not everyone has been able to spend as much time physically in the laboratory or physically in the clinical trial space, I think it has given us a little bit more time to kind of get inside our own heads and think a little bit about problems related to cancer that we've been addressing and potential new ways to do that. I think we've had an opportunity actually to potentially forge some new collaborations. So Dr. Gustafson and I have talked a lot about actually starting to do more sort of transdisciplinary or multidisciplinary collaborations. So a surgeon collaborating with a medical oncologist, a basic scientist collaborating with the surgeon, a pharmacologist collaborating with a medical oncology resident. So I think we've really sort of opened things up a little bit more and thought about ways that the laboratories and the clinical trials teams can interact a little bit more robustly, and I think that's going to potentially continue long term. We've also, again, had a little bit of time to sit down and do a bit more writing of grants, for example, looking for additional funding mechanisms for some of the ongoing research and new research moving forward, and I think that's been something that hopefully will come to fruition in the very near future. We're continuing to fundraise for OneCure, the clinical trials program endowment. And actually, there's been quite a bit of very positive success in that arena, and we're very, very keen on continuing to move forward with that initiative so that we can actually ensure the security of that program moving forward in perpetuity.
Can I just make a
Sure.
quick follow-up to what Doug says? This interface between the laboratories and the clinic is really what distinguishes CSU in the context of we have a very robust basic science program, we have clinical science, we have a clinical trials program. So what we're actually involved with now and have been building in pieces is a core facility that would take our clinical material, the archive material. Dr. Lana is in charge of the archive. These are tissue samples and biological samples. Essentially, that's the raw material from our patients that walk through the door with cancer. we build on a toolbox from our laboratory side, which is a way for us to understand the genetics, the biology of the cancers. We have cell lines that we've developed and studied for a long time, and some of those have come from those archival tissues. So we go from our raw material to our toolbox, and then ultimately to our overall workspace, where we have a clinical trials program that will allow us to implement studies that ultimately will lead to innovations and new developments. So this whole interface, this whole context of the translational core facility we're trying to build is really important for us here.
From the laboratory to the Labrador and back.
That's right.
I can see some of your faces on the screen up there, and I had a list of folks that were going to participate, but we want to thank you, first of all, for joining us. As many of you know, the state of Colorado would allow us to be good, but philanthropy, good friends, allows us to be great, so thank you very much. We've got some time for questions. 3 questions at least were submitted ahead of time. I think I can divide them up to the 3 panelists here. And then I think there's a way for you to ask questions real time here. We'll go on till 3 o'clock. So the first question that was submitted, this would be for Doug Thamm. What is the status and timeline of the vaccination against canine cancer study?
Great, yeah, thanks for that question. I'm not sure who asked it, but great question. So for those of you who aren't familiar with the study, this is an 800-dog multi-institutional, so this is actually taking place at several places around the country, randomized placebo-controlled trial that's looking at a vaccine, not necessarily a vaccine to treat an existing cancer, but maybe to prevent a large number of different kinds of cancers simultaneously. So we at CSU, University of California, Davis, and University of Wisconsin-Madison are all recruiting normal, healthy dogs between the ages of 5.5 and 11.5 to participate in this study. And they have to live within about 100 miles or so of one of those 3 participating sites. So no history of cancer, no autoimmune disease, no current treatment with any kind of immunosuppressive medication. Mixed breed dogs and certain kinds of purebred dogs are eligible. These dogs are very thoroughly screened to make sure that they're in good health to start. And then they're randomized simply to receive a vaccine or a phony vaccine. And they're checked twice a year for 5 years. At the end of the day, we want to know who got less cancer, the ones that got the real vaccine or the ones that got the phony vaccine. And there's a lot of safety things and immune system things that we're doing as well, so it's not quite as simple as it's made out to be. But at the end of the day, really, there's several deliverables that can be very useful from this study. So one is, obviously, there's the animals that are participating have a 50% chance of getting a vaccine that could delay or prevent cancer in their individual dog. We might end up with something at the end of the day that could be useful as a cancer preventative in dogs in general. And perhaps the most exciting component is this really would lay important groundwork for investigating a similar approach in people. And again, a vaccine to prevent cancer in people could be really game-changing. And we at CSU are incredibly excited about being involved in that research. So again, as I mentioned, the goal is about 800 dogs. And we just crossed the 400 dog mark in the last couple of weeks. And this is all 3 sites put together. At CSU, we're aiming for 285 enrolled dogs, and we just crossed the 150 dog mark. So we still have some work to do. We still need some patience. So if you guys out there happen to have a dog that falls into this category, and again, it's the right age, or if you know somebody who has a dog that falls into that category, if you're able to sort of broadcast this on social media, we're, again, actively recruiting more patients for this particular study. It's a win-win-win, I think, for everyone. We would love to have additional participation.
Thank you. The next question that came in was, for Dr. Lana, are there any trials currently ongoing for mast cell cancer in golden retrievers?
Maybe I can take that one too, and then you can do the third one.
Yeah.
So as things stand this very second, the answer is we don't have any ongoing trials for mast cell cancer. However, our director of clinical trials, Dr. Kristen Weishar, has submitted a research proposal to the American Kennel Club for a new protocol that will actually look at 2 existing drugs that both have activity in some dogs with mast cell cancer, and actually putting them together to see if that combination might be better than either one alone. So we should be knowing a little bit more about the status of that application toward the end of the year. So stay tuned for that. We may actually have some information about that in the next couple of months.
And there's also a sample collection study that's going on for mast cell tumors that is looking at a new marker to help predict which ones are more aggressive than the others that Dr. Weischer is involved in.
I can't pass up this opportunity to remind my medical oncology colleagues that 80 to 90% of mast cell tumors in dogs are cured by surgery
It's called
alone.
a scalpel.
Thank you,
Very
Dr.
true.
Withrow, for
Just-
reminding us.
I have to ground them in reality occasionally.
We know that.
And maybe for Dr. Page, but any of you, is there any research on nutrition to prevent cancer?
There's,
Go for it.
yeah, feel free. There's a lot of interest in that, and it's one of the more common questions that we are asked. There's no answer to that at this point. I think the nutritionists that I know that have asked questions similar to that will state that, you know, the best recommendations are to feed your dog a well-balanced, complete dog food, and that can be in any different format. In some cases, it's you know, raw diets or homemade diets or commercial diets. And there are some potential recommendations for supplements that might be useful. Most of the nutritionists believe that omega-3, omega-6 fatty acids in the right proportion are reasonable supplements that might be useful. There's also evidence for addition of vegetables to dog food, both green vegetables as well as other vegetables. But nothing specific that has been identified to prevent cancer at this point.
And I'll just add that now at CSU we do have a nutrition specialist and a nutrition service that's available for consultation. Her name's Dr. Camille Torres. She does virtual consultations and telemedicine consultations as well for individuals. So we haven't had someone on site with us before, so it's kind of a nice opportunity to treat the patient as a whole.
My minor editorial, there seems to be a lot of strong opinions and weak data when it comes to nutritional management or prevention. And it's a great area of study, and we need to probably continue to pour resources into that. So those are the 3 questions I had, and as I understand it, Lauren or Melissa are going to be able to forward questions to us that we can answer collectively.
Yes, we do have quite a few questions in the chat, so we're just going to see what we can get done here. The first question is, where can we find more information on environmental effects of carcinogens on companion animals? Something in general terms. I am not in the medical field.
Ron?
Well, so there's I don't believe that there's a really useful summary yet of these types of exposures that has been published to the extent that I would be able to direct you to that. I believe that there's information on a variety of different compounds. If you would like, I'd be happy to direct you towards specific papers or specific articles. But the workshop that I spoke about is going to result in a publication from the National Academy of Sciences that would probably be the best resource for this. And going forward, there will be a lot of awareness and campaigns. There'll be a lot of discussion about it. But both indoor, outdoor exposures to flame retardants or particulate matter or insecticides, herbicides, heavy metals, carbon pollution from cars, all kinds of things have been identified both in people and in dogs as a potential for concern and for monitoring. And there's been some really interesting ways of showing now that there are very similar exposures between our pets and ourselves. So I'm going to have you come back in about 6 months to a year and ask me the same question, and I'll be able to give you the answer.
Okay, did any students or animals test positive for COVID?
So I can answer that one. We'll talk about the animal part first, and there's some, there are animals that can be infected with COVID. I think what we know now, and that ranges, there's a variety of species, dogs, cats, mink. The larger animals, the equine and food animal types don't seem to be able to become infected, even in experimental situations. But what I think we know, and there are several people that do this type of research at CSU and then globally, is that it's pretty clear that humans cannot catch COVID from an infected dog or cat. We can pass the virus to our pet, but they can't pass it back to us. And so when this all started and we were seeing pets, there was a lot of concern that we could, that the animal would serve as a fomite, right? So that if a person had COVID and coughed it onto their dog, then the veterinarian would pick it up from their dog. And we know that that's unlikely at this point. And so while a pet could get it, the likelihood of them getting sick and or passing it is very, very low. From a standpoint of students and the rest of us who interact, we know that in the vet hospital, our biggest concern is for us to stay safe with each other. We are to maintain 6 feet distance at all times as best we can. We have taped off areas within our workspaces so that here's your place to sit with your mask on, 6 feet apart, here's your place to sit. We know that when we're working directly with animals together, for example, in surgery, drawing blood, things like that, we have to be closer than that. And so for that purpose, we all have to wear masks, we all have to wear eye protection or face shields. If we're within 3 feet of each other for more than 15 minutes or even within 3 feet for any length of time. And so we keep strict distances and guidelines as best we can. From a standpoint of positivity within the hospital, every employee or every person who comes into the hospital has to do a daily self-check where you take your temperature, you log any symptoms you have into the computer before you come into the hospital, and they will let you know whether you can come in or not that day. If you're sick, you're asked to stay home. From a standpoint of have there been positive cases that have arisen, there are positive cases that have been identified, whether they originated in the hospital or somewhere else. So a student or an employee may have been positive, and then contact tracing through the university comes into play. They talk to the people who work in that unit. They find out whether people have considered to be exposed, meaning they haven't had PPE on and they were near a person who tested positive for whatever length of time. And then you're asked to quarantine or not and be tested or not. So public health, both at CSU and within Larimer County, triggers many of the actions if someone has symptoms that are appropriate for COVID or has a positive test. So they do contact tracing for everyone. Within our unit, we have not had anyone that we're aware of that's positive. We've had people who've quarantined for other reasons or other family members that are positive and not been able to come into work. But everyone who can work from home does. Those of us on clinic duty are on the clinic floor, but everyone else, for the large part, if you don't have to be in the building, you're asked to work from home.
What % of your research is directed to cats versus dogs?
So great question. So this is something that's a little bit unique to CSU's population, but on the small animal side, and this doesn't just go for oncology, but it's actually the entire small animal hospital, about 85% of the small animal caseload at CSU is dogs. Only about 15% is cats. And that's actually reflected in the population for the oncology service as well. For that reason, as well as for some other reasons, we don't have as many tools to study cat cancer. We don't have as many cat cell lines. Really, a large majority of the research that is being done currently is on dog cancer specifically. So that being said, we do have an ongoing study. Actually, I take that back. We have two ongoing studies for cats as far as clinical trials go. So we have a radiation therapy study for cats with a form of oral cancer. And actually, we have what's called a pharmacokinetic study. So we have a study that's looking at the pharmacokinetics or the drug levels of actually two different commonly used drugs in cats. One is prednisone, and the other is a drug called chlorambucil, which is used for some kinds of cat cancer. So those three studies are actually ongoing right now, but on the basic science side, currently, again, a lot of it is really dedicated toward dogs because for one reason or another, that's most of what's coming in our front door.
Craig Webb's GI lymphoma
Yep.
work as well. So,
Yeah, there's a large effort to look at GI lymphoma or GI cancer from internal medicine specialists that has achieved somewhat of a national profile as well.
What is in the preventative vaccine, and why wouldn't this already be happening with humans?
Great questions. So
It's magic.
it's magic. So the preventative vaccine actually consists of a large number of different components. So each of these are a little tiny piece of protein that codes for what's called a neoantigen. So this is a new-looking protein to the immune system that seems to be present on a large number of cancers, both in people, mice, and in dogs as well. And the little hook with this, the reason that it's unique and different is that my colleague who's doing most of the scientific work on this, Dr. Stephen Johnston from Arizona State University, found a new way to look for these neoantigens that no one had looked for before and actually discovered a large number of them. So the vaccine that's being tested consists of about 27 of these little neoantigens in this vaccine. And they've been designed to target, kind of the 7-ish most common canine cancers that we see. So lymphoma, melanoma, mast cell tumor, osteosarcoma, soft tissue sarcoma, hemangiosarcoma, histiocytic sarcoma. So it's not pan cancer, every single cancer under the sun, but really trying to hit the majority of them. So why isn't it already being looked at in people? So that is a great question. So one reason is that this is a fairly paradigm shifting kind of approach, and a lot of people are skeptics about its ability to be successful, despite the fact that it looks incredibly promising in mice. So if you take any one of these little antigens or neoantigens and use them as a vaccine in mice, you can delay or prevent certain kinds of cancer. And if you combine them, the more you add, actually the better the protection is. So that's pretty compelling. But people who have done cancer research for a long time are the first ones to say, mice are not people. Just because you can make something go away or improve the outcome in mice really does not say very much about how it's going to work in people. And really moving this into a preventative setting in people is a massive, massive undertaking. You'd be talking about tens and tens of millions of dollars and 20 years probably to figure out if it worked or not. So one of the ways that we can take advantage of, and I don't mean that in a bad way, but utilize dogs as a model here, are the fact that, that old adage, so one human year is about the same as 7 dog years. That's pretty much accurate. So because of dogs' contracted lifespan, the average dog only lives to be 12 or 14, you can answer these questions about how well prevention of an old age disease like cancer works in a period of time that's probably 5 times faster than what it would take in people. So for the reasons of, again, being able to do it quickly, being able to do it somewhat less expensively than it would be on the human side, are really the reasons that the plan is to really start with start with dogs first. If the results look positive, then I think we've got a lot more ammo to go back and say, hey, look, this really does look like it has the potential to be useful. Here's some information that we've generated in real cancers, in real patients. How can you not look at this in people? So that's actually the plan and the reason why we're starting with dogs first.
We have a lot of great questions. I'm afraid we're not going to get to all of them today. I'm going to ask 2 more questions. those people who didn't get their questions answered, we will be following up with you and providing more specific information. The next question I wanted to put out to you has to do with osteosarcoma. And are there any exciting developments recently in osteosarcoma?
Who wants to answer that?
Go for it.
So from a treatment standpoint, our therapies haven't changed a whole lot from a standpoint of you treat the primary tumor and then you provide chemotherapy. Stereotactic radiation is something we've been doing for a long period of time that allows a limb to be saved for those patients who amputation is not a good choice for them. Surgical limb sparing techniques are continuing to evolve and change. I think we're getting a lot better about being able to select which therapy might be best suited for which patient. I think the place where we need to focus more work and more basic science, and maybe Doug or Rod can jump in here, is once we have metastatic disease, what can we do? And are there other approaches that we can turn to? And so there are a couple of clinical trials that are ongoing to try to prevent or slow down the metastatic disease process. And then we have a couple of trials that also are looking at, if you're already developed metastatic disease, how can we maybe harness the immune system or do a better job at being able to slow that process down?
Yeah, so I can jump in. So Dr. Steve Dow, who's a tumor immunologist on our faculty at the Cancer Center, actually is the recipient with his colleague Cheryl London at Tufts of a large NIH grant, specifically looking at novel therapies to try to delay, prevent, or treat metastasis or spread in osteosarcoma. And he and Dr. London are actually testing in small-scale studies a number of different approaches with the idea of whichever one of these looks the most encouraging in these small-scale studies, they're going to pick and then actually utilize in a much larger study to try again to prevent rather than treat clinically relevant evidence of spread. Another thing that I guess I would mention that I'm very proud of is the fact that we're one of the founding sites for participation in a group called the Comparative Oncology Trials Consortium. So this is a group of what's now probably 28 veterinary schools around North America that are coordinated by the National Institutes of Health, the National Cancer Institute, with the idea of doing large or complicated clinical trials to inform not only the treatment of canine cancer, but the treatment of human cancer as well. And over the years, we've actually been involved in a series of studies looking at novel treatments for osteosarcoma. And the results of a lot of those studies are not public yet, but over the years, we've probably enrolled 100 dogs or more in these studies that are sponsored by the National Institutes of Health, and again, a product of collaboration around the country.
I'm going to let this last question actually let the questioner ask the question. So we're going to unmute Jeff New, our friend Jeff, and let him ask his question.
Which question was I asking? Hold on. Oh, how do you test dogs for COVID? The same way as humans?
Yes.
So I can just have a regular test on them?
I don't know that a lab that's testing humans would accept your sample, but
Why?
I think it's the same technology because they may not accept animal samples.
So if I told them it was me, we could just do it.
I didn't hear that.
Yeah, me either.
No, I said if it was, I just told them it was me, they could test it.
They should be able to, yes.
The only
I
thing that
don't think
wouldn't-
there's any difference.
I say, so if you're doing one of the nasal swab tests, the answer is yes, because those are either looking for the genetic material or looking for the protein that's made up by the COVID organism. An antibody test wouldn't work.
No.
So you couldn't do that, but if you were doing an antigen test or the genetic test, theoretically, I ain't telling you to do it, I ain't telling you to lie, but those would be work.
Thank you. Good, because I wanted to have Charlie tested after deployment. I wanted to make sure she was COVID-free.
So they've tested animals.
Yeah,
Yeah.
they
But it's
do,
done at veterinary diagnostic labs, not
Right.
human, so.
yeah. But I can tell you, if you try to stick that little swab up your dog's nose, you're probably not going to be your
won't
dog's
be happy.
favorite person anymore.
I'll let you do it, Dr. Lana.
Okay, thanks. Happy to.
Thank you, everybody.
Okay, well, thanks everybody for participating in this. Hopefully we got some positive information out for you. Again, I'm really proud of how our team has risen to this crisis and how we've changed operations to optimize patient care. So thanks everybody. If there's additional questions, you can run them through Lauren and we'll get those questions all answered. So I'd like to thank Lauren Mingus and Melissa and all the CSU staff for putting this on. Much bigger deal than you guys can imagine at the other end there, but it's worked out great. So thanks very much.
Thank you all.
Bye.
Thanks a lot.
Thank you.